Hello, how are you? Good. How was your Thanksgiving?
Good.
Yeah? What'd you do? Anything?
Sunday to technically Sunday,
Okay.
all playdates. The playdates.
You did what?
Dinners, playdates, all.
Oh, play dates. Okay, dinner and play dates. All right, that's cool.
I didn't take a break. And
Wow, you were busy. Okay, well, that's cool. That's good. Rather be busy than bored, I always say. You know, this is true. I would rather be busy than bored. Well, that's fun. Did you have turkey?
yeah,
Awesome. Yeah. Did you have a lot of family or friends over, or just your family?
friends.
Friends. Okay, very cool. Very good. And we had friends as usual. And one of our friends brought a new friend from Peru, and this was the first time he'd ever done Thanksgiving. So we got to be his first Thanksgiving, which was kind of cool. Real nice guy. Enjoyed getting to meet him. Hopefully he'll be around more because he was cool. He was fun. He just fell right in, comfortable with everybody.
Third grade teachers.
For Peru? Okay, there you go. Hello, Miss Everly. Hey, Emerson. How was y'all's Thanksgiving? Was it good? Yeah? Did you have a lot of family or friends over, or did you eat at your house, or did you go someplace, or did you guys host dinner? You don't know. We are unable to speak again. Okay. Well, cool. Well, I was saying we had some friends over here, and one of them brought a new friend who's from Peru. So that was kind of cool. Yeah. Excellent. All right. Well, we'll go ahead and start since we're a couple minutes behind. I've got my glasses on today. I think I scratched my cornea in my right eye. It's really bothering me. So I considered canceling today because sometimes I'm having trouble keeping it open. So anyway, we're gonna roll with it as best we can and then probably have somebody look at it later today if I can. All right, so we have this lesson, right? And then one more lesson, and then we're in midterms already. Can you believe it? That was fast, right? Halfway through the year almost. So we'll go ahead and start with lesson 15. So we're talking about population evolution today, not individual and evolution on an individual basis is what leads to population evolution. But population evolution is the important thing to consider when we're looking at change over time. So let's talk about fossils and what kind of evidence of evolution can you find in fossils? So you've got fossilized plants and animals, and they can serve as evidence of evolution. So what kind of evidence? How? So scientists use fossils to find similarities between ancient species and the ones that are alive today. So we look for things that are similar. The location of fossils on Earth, how deep underground they are, can tell us a lot about how the species lived and how long ago it was alive. And it can also tell us a lot about the Earth too, like where we find fossils. Like we talked before, I think about sometimes you can find marine animals up in the rocks in the mountains, and that tells you what? What would it tell you if you found a fossil of a marine animal up in the mountains? Yeah, that it used to be ocean. So we expect an ancestral species to share some common traits with modern species. So if a modern species arose from an ancestral species, we're going to expect to see some things in common. So living things from the past are different than living things from today. Now, why do you think I chose this particular picture? Why do you think?
They're related.
They are absolutely related. Some of our dinosaurs that we know best eventually evolved into modern day birds. So they are related. That T-Rex and that tiny little chick are related. So I'm going to move this thing for a minute. Whoops. Move this for a minute so you can see up here at the top what we're dealing with. So these are embryos, all right? And these are embryos at different stages of development. So up here at the top are the earlier stages. These are kind of middle, and these are later stages. So you have a fish, an amphibian, a reptile, a bird, and a mammal. Now, I want you to look at this line here at the top of those 5 embryos and tell me something you see in common in all 5. What's something you see, Everly? They're all pink. Okay. Well, how about structure? What do you see? A tail? Is that what you're looking at? Yeah, they all have a tail. The fish has a tail. The amphibian, the reptile, the bird, and the mammal all have a tail. Do you see anything else, Evan? I see eyes. They all have eyes. Do you see that? And then as you move along, you can see they start to differentiate more and they become more dissimilar with time, right? You can see the mammal still has a bit of a tail here. What mammal is that, do you suppose?
Human.
Yeah, it's a primate of some kind, either a human or maybe a chimpanzee or something like that. But that's definitely a primate. Looks human to me. This looks like, any kind of bird you could see. They all look pretty similar, right? So, and this guy is probably some sort of a salamander. You see, he's got these little external gills going on. So vertebrates develop in similar patterns. So over time, you see that these vertebrate species, they all kind of started similarly and then they differentiated as they went along. But the patterns of development are similar. And if you look at this image down here, it's showing you these color blocks are showing you how those same structures are organized in different organisms. So these, I would call this cervical vertebrae. That's like your neck, right? So you can see how it's different there from there to there, but it's the same set of vertebrae. And then look down their spine. They've got thoracic and then lumbar vertebrae. And you can see how they're the same kind of structure, but it's developed differently in each of these organisms. All right, now we have to talk about analogous and, oh gosh, I'm gonna get that out of my way today. Arrangement of bones shows how organisms may be related, and then some features have similar internal structures but serve a different purpose. So we have analogous structures and homologous structures. Does anybody know the difference between an analogous structure and a homologous structure? So a homologous structure is similar in form, but not necessarily function. And an analogous structure is similar in function, but not necessarily form. So here's a great example of that. Let's look at the butterfly wing and the bird wing. What is similar, their form or their function? What do you think? What's more similar? The function. They're for flying, right? The butterfly wing is for flying, the bird wing is for flying. But their function is similar, but their form is very different. There's no bones in that, right? Are there bones in a bird wing? There's bones in there, right? So if you look over here, here's homologous structures. So in this case, they've got a similar form, but their function may be different. Does a bat use its forelimb differently than a human? This is your forelimb. How does a bat use its forelimb? What's it do with it? It flies with it, right? And we don't fly. Our forelimbs don't. I wish they did, but so they don't. So if you look at the bones, they've highlighted where those same bones are positioned differently. So homologous means you have a similar form, and analogous means you have a similar function. Okay? All right, so let's keep looking. Oh, wait a minute. I didn't mean to hit that button. Go away. Okay. So here you can see how horses have evolved over millions of years. So if you look right there, you can see that little 60 million years ago, we had that little organism. And you can see how the bone, look at the bone structure in the leg. You see how it's evolved over time? It's the same bones, but they've kind of changed position as the animal changed. So why is this keep going up so high today? All right. So fossils of the simplest organisms are found in the oldest rocks, and fossils of more complex organisms are found in the newest rocks. Why do you suppose that is? The simplest organisms are found in the oldest rocks, and the most complex organisms are found in the newest rocks. Why do you think, Emerson? Yes, because animals perfect. That's a perfect answer. Animals got more complex over time. And not just animals, but plants too. All organisms have gotten more complex and more specialized over time. So when we find newer rocks with fossils in them, they tend to be more complex organisms, whereas the older rocks tend to be the very simple or more basic organisms. Okay, why is that popping up? Go away. So this supports Darwin's theory of evolution, which states that simple life forms gradually evolve into more complex ones. And by studying fossils, scientists can learn how much or how little organisms have changed as life has developed on Earth. Now, there are gaps in the fossil record because many of our early forms of life were soft bodied, which means we have few traces left behind. So, if an organism is soft bodied, it doesn't have bones to stay behind to become fossilized, right? So, like, what's an example of a soft bodied organism you can think of? Something with no bones. A worm, that's a good example. Or maybe a jelly, like a jellyfish, a comb, a jelly, one of those. Yep. Those guys can be soft bodied. So we lack a lot of fossil evidence for those species because they don't preserve well over time, right? And then similarities in structures such as backbones can be observed in different organisms, which we just looked at. And then the same structure in ancestral organisms can show evidence of change over time. So if you see the same structure, like think back to those horse legs, the bones on the horse's leg, you can see how that structure changed over time. So now let's talk, you finished fossils, so let's talk about related species. What does it mean to be related? All right, so we have, bless you, we have comparative anatomy and comparative embryology. So this is embryology over here, and that's we just looked at those embryos a minute ago, and you said, oh, they all have tails, or they all have eyes, or they're all pink, you know. So let's look at, here's comparative embryology, and then this is comparative anatomy. And I have to say, I think the most fun class I ever took in college was comparative vertebrate anatomy. Not an easy class, but a ton of fun. So look closely here. It says that top animal, it says it's terrestrial. What does that mean to be terrestrial? What does that mean? Do you know what terra is in Latin? It's earth, land, ground, earth. So a terrestrial organism would live where, do you think? On the ground, on the earth, not in the water, right? And then you see the others say they're aquatic. So those are organisms that live predominantly in water. So when you have your terrestrial organism at the top, you see how he's got two sets of legs and then he's got a long tail. But if you look down here at the whale, do you see what it says right there? It says pelvis and hind limb. Did you know that whales, even though they have no rear legs, they still have a little pelvis where legs would be, right? So this just shows that these organisms are all related because they all have that backbone, they all have that spine. So they do have a common ancestor, but some structures are going to change over time. And if you look at the hearts, this is a really good example too. How many chambers do you see in that fish heart? I only see one. Do you see only one big chamber inside? And the amphibian, how many do you see? There's actually three, but you can't, yeah, it's hard to see because it's not 3D. But look at the bird and the mammal. There, we have four by the time we get to birds and mammals. So even things like circulatory systems are going to evolve. It's not just bones, it's everything. And then if you look over here at these little critters, you can see how embryology is compared. They all look similar. They start to look more different. They still look pretty similar, but they're starting to change. And then you get to the point where they're very differentiated and specialized. All right, so comparative genomics. So this is interesting. Now what we can do, because science has gotten so advanced, we can compare DNA in organisms to see how they are related. How much DNA they share in common tells you how closely related they are. Think back to your cold case lab. You were looking for people in the database that shared the DNA with your suspect, right? And the more DNA somebody shares, if you remember the centimorgan chart that was in there, the more DNA someone shares with another person, the more closely related they are. So the same can be said for different species. The more DNA that species have in common, the more closely related they are. So researchers chose the appropriate timescale of choose the appropriate timescale of evolutionary conservation for the question being addressed. So what that means is there are common features for different organisms, such as humans and fish, and that are often encoded within the DNA, evolutionarily conserved between them. Means it's as the animal continues to evolve, as organisms evolve, certain amounts of DNA don't change. Certain things don't change. So let's look at this little chart at the bottom. Now, I'm gonna add, y'all are siblings, so this won't work as much for you. But like Emerson and Evan, what percent of your DNA do you think you have in common? 0.1%, so almost nothing in common. What do you think? How similar? Like how how like is it a high number or a low number? How much do you think you share? How much DNA do you think you share of your whole genome? How much do you think you share? Don't know. What do you think, Emerson? A lot, a little? Do you think you share more with Everly or with Evan? Right, because you're related, right? Okay. But here's the crazy thing. So here's a chimpanzee, right? How much DNA do you think humans have in common with a chimpanzee? What do you think? 80%? Good guess. What do you think, Emerson? Higher or lower than 80? Higher than 80? All right. What do you think? A little higher? Okay. So humans and chimpanzees actually, why did it pause my screen sharing? Your screen sharing is paused. Why is it doing that? Share it. Oh my gosh, hold on. It's being, yeah, it's just being silly. Wait a minute. Where's the resume?
That's the chair.
No, hold on a second. It's being silly. Okay. Sorry, it's just being stupid. And like I said, I can't see very well today. Okay, there it is. All right, sorry, I couldn't see it. All right. Humans and chimpanzees can share 99.6% of our DNA. Now we know DNA is used to encode for proteins, right? DNA makes RNA, RNA codes for proteins. What percentage of proteins do you think we share with chimpanzees? Anybody want to guess? We share 99.6% of our DNA with them. We actually have 100% of the same proteins. They produce the exact same proteins we produce, 100% across the board. How about a dog? Less or more than their 99.6? Everybody says less? Everybody agrees? Less? Why do we say less? Why? Okay, so we're not in the same order or genus, maybe is kind of what you're getting at. Yeah, we're not. Okay, they're a different type of animal. They're a canine, we're a primate. Okay, you'd be right, but we still share 91.3% of the same DNA that dogs have, and 95.2% of the same proteins. Now, how about a fruit fly? A tiny little fruit fly that comes home from the grocery store and invades your fruit basket. How much do we think? What percent? 70? Okay, good guess. 60, he says. All right. So 72.4 of our DNA is the same as that in a fruit fly. And our protein similarity is 76.7%. So you notice that the protein and the DNA numbers are pretty close, right? That makes sense, right? Because our DNA codes for mRNA and our RNA makes our protein. So how about a little roundworm, a little itty bitty tiny, very basic, lower level life form roundworm? What do we think? That one's going to be 68%. So still way more than half, right? And 74% of our proteins are the same. So that's kind of wild, right? All right. So think about it. What do we analyze to study evolution at the molecular level? What do you think? Our genomes, right? All of our DNA, right? DNA evidence may indicate how two species are related to one another, even if their body structures don't offer clues. A fruit fly is not going to offer too many clues how it's related to us, right? Aside from the fact that maybe they have limbs and segmented bodies and eyes. Like that's about it, right? So we have all kinds of relationships here that we can diagram. These are all bacteria and how they're related. I want you to notice this though. We've got some domains here. We've got bacteria, archaea, and eukarya, right? And I want you to notice the common ancestor is way down here. It does not mean that bacteria evolved from archaea, evolved from eukarya, or that that eukarya evolved from bacteria or archaea in any way. It doesn't. It's a different branch. You see how it branches off entirely? It's not a straight line. It's a tree. And over here, this shows how different bacteria are related as well. So now let's talk about speciation. Can anybody name all these? I can, because I love my birds.
I only know the
You know the cardinal? So we actually have two cardinals. That's a male cardinal, and that's a female cardinal. That's a goldfinch. That looks like an eastern towhee to me. And that's an eastern bluebird. Yay. I love my birds. I love to watch my birds. So, what is a species? So, a species is a group of living organisms consisting of similar individuals that are capable of exchanging genes or interbreeding. And they can mate and produce offspring, but there's something specific about the offspring. Does anybody know what it is? We talked about it a little bit before. Remember the zonkey? What was the zonkey?
A donkey.
It was a zebra and a donkey, right? It was a cross, a hybrid cross. Are zebras and donkeys the same species? No, but they have the same number of chromosomes and their genes line up appropriately so they can produce this hybrid, just like horses and donkeys produce mules, right? But those animals, they cannot reproduce. That offspring cannot reproduce. So in order for something to be the same species, when they mate, their offspring has to be fertile. That offspring has to be able to reproduce. That's why a zonkey is not a new species. It can't. A donkey and a zebra are not the same species. So the zonkey will never have babies. The zonkey cannot reproduce. It's just not something that works.
What if the donkey is donkey?
Nope, they're not fertile. They don't have the ability to reproduce. But that's a great question. So then there's a really pretty critter. This is a spotted lanternfly, and I just think they're beautiful. Okay, so the process of speciation. Somebody define speciation. What does that mean? Speciation. What do you think it means? So it means that you formed a new species, speciation. As new species evolve, populations become reproductively isolated from each other. When members of two populations cannot interbreed and produce fertile offspring, we call it reproductive isolation has occurred. Now, this is important. You want to isolate your group so that you can get a new species. So when populations become reproductively isolated, they can evolve into two separate species. Reproductive isolation can develop in a variety of ways. Does anybody know? There are three main ways that we get reproductive isolation. We can do it through behavioral isolation, geographic isolation, or temporal isolation, and we'll talk about those. And I like our little pigeons. He says, what are other words for speciation? And this guy says biological evolution, convergence, parallel evolution, adaptive evolution, evolution, organic evolution, and phylogenesis. Those all mean roughly the same thing. We have lots of words, don't we? So let's talk about reproductive isolation and how it works in different ways. So we'll talk about behavioral isolation. This is one of our three ways, right? In behavioral isolation, you've got two populations that are capable of interbreeding, but have differences in maybe their courtship rituals or other behaviors that make it so that they don't interbreed, even though they're capable of it. Sometimes social animals develop courtship rituals. You ever seen the bird? Like the bower, what is he, the bower bird, the guy who like he builds this fancy nest and then he brings the ladybird over and he like dances for her. You ever seen that? You ever seen the birds that dance, like the male birds that dance to try and attract the female birds? They look crazy. You can look up videos of them. Some of them are really funny. But that is a courtship ritual. So if for some reason that's, it's appealing to a certain part of the population, but not another part of the population, that's a behavior that's going to isolate the group that's dancing a certain way, maybe from a different group. And that can lead to speciation because they're only going to breed with each other over here and they're only going to breed with each other over here. Then you have geographic isolation. This one's really easy to explain. Geographic barriers, such as rivers and oceans and mountains in some cases. Look at the creatures that live in Australia. Do we have creatures like that anywhere else in the world? They're very different, right? They're very, very different because they evolved in that isolated island, basically, continent, but it's a small continent, right? Big island, small continent, away from everybody else. So because of that, they've been isolated, and that interbreeding is not going to mingle with any other organism any other organism of the same species that might have different genes mixed into the equation. And then lastly, we have temporal isolation, and this occurs when two or more populations can interbreed, but they only reproduce at different times. So certain animals only reproduce at certain times of the year, right? Humans, we get 12 opportunities in a year to produce an offspring. Some animals, like dogs, maybe get like 2 opportunities in a year to produce an offspring. And sometimes it's related to seasons, like deer, they will rut or mate in the fall, and then their moms are pregnant throughout the winter, and then the babies are born in the spring. But if for some reason that change, that fertility cycle changed, then you would have animals that would not be mating with one another because their mating cycles are not aligned. All right, so then also notable, what's a group of animals of the same species that mate and produce offspring? This was way back when we talked about ecology. That's a population, right? And what is it when we have all the genes, including all the different alleles in a population? What do we call that grouping of genes? Do you know? All the genes. We call that the gene pool. Okay, so all the genes, all the alleles, we call a gene pool. Now, researchers study gene pools by examining, what do you think about alleles? So this one is relative frequency. All right, so what this means is how often does the allele for this particular characteristic appear out of all the alleles? How often does this one allele appear out of all the alleles? So the relative allele frequency is the number of times a particular allele occurs in a gene pool compared with the number of times others occur. All right, how many frogs? We have 15. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. How often does a red frog occur? 1 out of 15. Perfect. So the relative frequency of that would be 1 out of 15 or 1/15 of the time. And you could actually make that a percent by dividing the 15, the 1, right? How often does the purple frog appear? 2 out of 15 times, right? Okay, so this is the relative frequency. How often does the green frog appear? 12 out of 15. So that is the relative frequency of the allele. How often do we see it? And then you can see it in the gene pool, the same thing, right? All right, evolution is any change in the relative frequency of what in the gene pool? What were we just talking about? The relative frequency of alleles in the gene pool. Natural selection operates on individuals, but resulting changes in allele frequency show up in populations. So it's populations rather than individuals that evolve. So say you had a population of frogs that was 80% for green genes and 20%, sorry, for brown genes. And then after a year, you got 60% for green genes and 40% for brown genes. There is evolution in the population. Individuals change, but it's the population that's going to evolve. That's what matters in the long run. All right, so let's look at how genes are shuffled. Why am I getting texted? Yeah. We go to Philly, not so much, but visit Philadelphia. Okay.
The 5 fingers of evolution. A thorough understanding of biology requires a thorough understanding of the process of evolution. Most people are familiar with the process of natural selection. However, this is just one of 5 processes that can result in evolution. Before we discuss all 5 of these processes, we should define evolution. Evolution is simply change in the gene pool over time. But what is a gene pool? And for that matter, what is a gene? Before spending any more time on genetics, let us begin with a story. Imagine that a boat capsizes and 10 survivors swim to shore on a deserted island. They are never rescued and they form a new population that exists for thousands of years. Strangely enough, 5 of the survivors have red hair. Red hair is created when a person inherits 2 copies of the red gene from their parents. If you only have 1 copy of the gene, you won't have red hair. To make this easier, we will assume that the 5 non-redheads are not carriers of the gene. The initial frequency of the red hair gene is therefore 50%, or 10 of 20 total genes. These genes are the gene pool. The 20 different genes are like cards in a deck that keep getting reshuffled with each new generation. Sex is simply a reshuffling of the genetic deck. The cards are reshuffled and passed to the next generation. The deck remains the same, 50% red. The genes are reshuffled and passed to the next generation. The gene pool remains the same, 50% red. Even though the population may grow in size over time, the frequency should stay at about 50%. If this frequency ever varies, then evolution has occurred. Evolution is simply change in the gene pool over time. Think about it in terms of the cards. If the frequency of the cards in the deck ever changes, evolution has occurred. There are 5 processes that can cause the frequency to change. To remember these processes,
This is great.
we will use the finger on your hand, starting from the little finger and moving to the thumb. The little finger should remind you that the population can shrink. If the population shrinks, then chance can take over. For example, if only 4 individuals survive an epidemic, then their genes will represent the new gene pool. The next finger is the ring finger. This finger should remind you of mating because a ring represents a couple. If individuals choose a mate based on their appearance or location, the frequency may change. If redheaded individuals only mate with redheaded individuals, they could eventually form a new population. If no one ever mates with redheaded individuals, these genes could decrease. The next finger is the middle finger. The M in the middle finger should remind you of the M in the word mutation. If a new gene is added through mutation, it can affect the frequency. Imagine a gene mutation creates a new color of hair. This would obviously change the frequency in the gene pool. The pointer finger should remind you of movement. If new individuals flow into an area or immigrate, the frequency will change. If individuals flow out of an area or emigrate, then the frequency will change. In science, we refer to this movement as gene flow. All 4 of the processes represented by our fingers can cause evolution. Small population size, non-random mating, mutations, and gene flow. However, none of them lead to adaptation. Natural selection is the only process that creates organisms better adapted to their local environment. I use the thumb to remember this process. Nature votes thumbs up for adaptations that will do well in their environment and thumbs down for adaptations that will do poorly. The genes for individuals that are not adapted for their environment will gradually be replaced by those that are better adapted. Red hair is an example of one of these adaptations. Red hair is an advantage in the northern climates because the fair skin, allowed ancestors to absorb more light and synthesize more vitamin D. Thumbs up. However, this was a disadvantage in the more southern climates where increased UV radiation led to cancer and decreased fertility. Thumbs down. Even the thumb itself is an adaptation formed through the process of natural selection. The evolution that we have described is referred to as microevolution because it refers to a small change. However, this form of evolution may eventually lead to macroevolution or speciation. Every organism on the planet shares ancestry with a single common ancestor. All living organisms on the planet are connected back in time through the process of evolution. Take a look at your own hand. It's an engineering masterpiece that was created by the 5 processes I just described over millions and millions of years. Can you recall the 5?
Okay, so can we do the 5? What's the little one stand for? Populations may shrink, right? Populations can shrink. What's the ring finger stand for? Non-random mating or selective sexual reproduction. What does the middle finger stand for? Mutations. And then what's the pointer stand for? Movement. Organisms can move in and out of an area. And then what's the thumb? Adaptations are either selected for or against by nature, right? That's a great way to remember it. All right, so the last thing we're going to watch here is about Hardy-Weinberg equilibrium. And I don't want you guys to get too anxious over it because you're not going to be required to like really deeply understand it, but I want you to know about it because it really explains some things. And it's mathy. It's mathy. It's a little basic algebraic-y, but it's good to know. Very good to know. And I know that, high school biology will require you understand it. So it's a good thing to look at.
My energy bill dropped from $150
Yeah,
to $50
yeah.
by doing this. My friends laughed at me when I spent $49.
Like math. Sometimes when people are starting out in biology, for some reason they assume there is no math in biology, but then they soon find out they were wrong. My math teachers were telling the truth when they said math isn't everything. Chi-squares, osmotic pressure calculations, standard curves with gel electrophoresis, Punnett square ratios, these all have math. But one of my favorite topics that involves biology and math, and is also absolutely fascinating, is the Hardy-Weinberg equilibrium. It got its name from a mathematician and a physician, and it states that a population's allele and genotype frequencies are constant, unless there's some type of evolutionary force acting upon them. A reminder from our ecology videos, a population in this case is a group of organisms that are all the same species and can breed with each other and have fertile offspring. However, there can be variety among a population as they're not clones of each other. But based on Hardy-Weinberg equilibrium, the population's allele and genotype frequencies would remain constant if no evolutionary force acts upon them. If we have these frogs here, assume they are all the same species in this example, but there are some slight differences. In our simplified example, some of the frogs are lighter green and some are darker green. We're going to use a simple genotype in this example. All frogs in this population will be either big G, big G, little g, or little g. There is also an allele frequency in our population. We're going to say here that 60%, a frequency of 0.6, of the alleles are big G. 40%, or a frequency of 0.4, of the alleles are little g. Notice the percentages add up to 100%, and the frequencies add up to 1. Now, to be in Hardy-Weinberg equilibrium, we have to have 5 assumptions. Number 1, no selection. No natural selection is acting upon these frogs. That means neither dark green nor light green will have any impact on reproductive fitness. Number 2, no mutation. Baby frogs inherit genes from their parents and there are never mutations. Number 3, no migration. Frogs can't come in, frogs can't go out. Number 4, large population. There are a lot of frogs. It turns out small populations are more vulnerable to genetic drift, by the way. See our genetic drift video. Number 5, random mating. The frogs mate without any specific choice. All of these 5 assumptions must be kept in order for Hardy-Weinberg equilibrium to happen. So in real life, does this generally happen? No. For example, in real life, maybe in a certain environment, predators can more easily see the light green frogs, so perhaps they are eaten more and have less reproductive fitness. So if Hardy-Weinberg equilibrium is unrealistic in nature, why does the Hardy-Weinberg equilibrium matter? That's where the math comes in. The Hardy-Weinberg equilibrium gives you this baseline to compare how an evolving population could compare to one that remains constant without evolutionary forces acting upon it. And now let's explore the math. So there are two equations in Hardy-Weinberg equilibrium that we'll focus on. We're going to start with the first one. P plus Q equals 1. In this equation, P is the dominant allele frequency in the population, and Q is the recessive allele frequency in the population. By the way, Hardy-Weinberg equilibrium doesn't mean that P has to equal Q, and the dominant allele frequency in any population doesn't have to be larger than the recessive allele frequency in a population. That's a misconception because dominant alleles aren't always the more common allele. The equation does say that the dominant allele frequency and the recessive allele frequency have to equal 1. So in my example here, if I say 60% of the alleles are big G, 40% of the alleles are little g, 0.6 is P, and 0.4 is Q. So this equation, P plus Q equals 1, is for those allele frequencies. But what if I wanted to know the genotype frequencies? Meaning, I wanted to know the frequency of frogs that are homozygous dominant, which is big G, heterozygous, which is big G, little g, or homozygous recessive, which is little g. Then I can use this other Hardy-Weinberg equilibrium equation. P squared plus 2PQ plus Q squared is equal to 1. So P squared is the homozygous dominant frequency, the frequency of big G in this case. 2PQ is the heterozygous frequency, so big G, little g frequency in this case. Q squared is the homozygous recessive frequency, so little g frequency in this case. Let's plug those previous P and Q values.
Have they lost you
Q
yet?
squared then be 0.36.
Skip ahead a little.
With that
This is where it gets
information,
important.
solve all genotype frequencies and all allele frequencies if in Hardy-Weinberg equilibrium. Step 1, determine first whether you're going to work with the first equation or the second equation. Since I'm working with individuals here that have genotypes, I'm going to work with the second equation first. Step 2, figure out what value you can determine.
So anyway, not going to make you guys watch the whole thing because you're not going to use it.
I can take care of
But
that.
want you to know that it's a thing and you can use it to determine allele frequency, which is important when it comes to population evolution. So know that there is a math equation out there that can be used. And why is that not clicked? I am. There you go. So it's a thing and it can be done and it's pretty darn cool. But you guys are going to have to do it.